We are longevity Genie team, and we are submitting to sequence to function hackathon track. For us, it's sequence to function to interaction, because interactions do matter. Genes can behave synergistically, extending lifespan together even more than separately, but more often genes can behave antagonistically, negating effects of each other. Also, not only type, but also place of interaction matters. For example, when you port the protein from long-living species into short-living, willing to extend its lifespan, you can accidentally break some interfaces between proteins it interacts with and get even to negative effect. So what is our approach? We obviously start from resolving the most important information about proteins and their relations to longevity using model context protocol servers that we developed previously. However, we are also using Atomica graph neural network model to efficiently and quickly characterize the interactions between the proteins and score them. For this hackathon, we released a custom fork of Atomica model. We pre-computed the dataset with longevity protein embeddings to let AI agents use it faster. And we made an MCP to glue it together and also to predict residues that are the most important for the interaction and that we have to take into account when optimizing the protein. How it all works together, Livia will show with the demo.
This is a demo of our solution. As you can see, we're having a query about a gene to know its sequence and functional orthologs, to find key variants and structure, and also to find complexes with key interaction partners. This is being presented via Claude Desktop using our own custom MCPs, since Claude does not have default information about like protein structures or longevity. Well, you can see the query live, and we're going to see how it outputs the results after querying all these databases in a draft format, like a markdown file, which will be displayed next to it. This is what they call an artifact. To be noted that this file can be taken further in a processing pipeline using the information within it. As you can see, you have several variants: functional impact, longevity, relevance, all structured nicely and easily to be found. This case is a case of searching from just a query, no previous files, and we'll use in the next part of the demonstration this file to be extended or to contribute basically to the next query. You have also sources. Here is the demonstration about the atomica-mcp. In this case, it searches for structures and it computes it computes the scoring basically. So it searches the UniProt database, then computes the values of scoring, and you have the output of the results here of the residues, obviously, Atomica critical residues. Novelty prediction, glycation analysis, and key insights in glycation and aging. So it's important to note the longevity and functions and interactions contributions that our MCP provided while using, well, a standard MCP client like Claude, but it can also be any other client you prefer.